My study was called Transgenerational Gene by Environment Interaction in Distinct Strains of Morphine-Exposed Mice. My mentor is Dr. Elizabeth Burns. She couldn't be here today. She's presenting at the Society for Neuroscience conference in Chicago right now, but I know she'll probably watch the video, so thank you to her. Also, thank you to Dr. Anika Tori, who was the postdoc student working in the lab, who basically held my hand through 2 summers of research. She was amazing, and she is now a full-time professor at Rhode Island College, so congratulations to her. The goal of my study was to determine the effects on offspring of maternal opioid exposure, even in the absence of in utero exposure. So that's a lot of jargon. The easiest way to explain it is to look at the timeline of the study. So the top bar is actually the maternal generation, and because it's transgenerational, the bottom line is the timeline of the offspring. So you can see from the top line that this blue region here is where the mothers were treated with either saline or morphine. Saline is just a water salt solution. It's just a control. And then you can see that for the rest of the mother's lives, they were drug-free. And there was a period of about 25 days before they were mated while they were drug-free. And then you can see from the bottom line that the offspring received saline or morphine when they were in adulthood. So they too lived their whole lives without being disturbed, no drugs, nothing, until they're adults, and then they're exposed to saline or morphine for 5 days. And the reason we have our study set up like this is because we're looking at gene by environment interaction in a transgenerational study. Gene by environment interaction is another way of talking about epigenetics. So for a long time, people believed that the only way traits could be passed down in the genome was natural selection or mutations. And so we know now that that's not true. A lot of times heritable changes can be much faster than that. And it can happen by mechanisms like DNA methylation or histone acetylation. And so we're looking at how saline or morphine exposure in mothers prior to in utero exposure translates to offspring. So this is the design of the study. There's lots and lots of different factors in this study. So first things we looked at were 2 different strains of mice. Then we have the maternal exposure. So the mothers either received morphine or saline. And then we have the offspring exposure. So all the offspring either received morphine or saline. And then it's split up by sex. So all of these factors lead us to have 16 experimental groups. And I mentioned briefly that we use 2 different strains of mice. First of all, the reason we look at mice is because 1, they reproduce really fast, so transgenerational studies are good when using mice. Secondly, they're cost effective. You can have lots of them and look at lots of different mice in a relatively cost effective way. But third of all, their genome and their phenotypic behavior surrounding addiction and withdrawal are pretty similar to humans. And the 2 different strains of mice that we use have longer names, but for the purpose of this study, we're just going to call them the 129s and the C57s. And they're different because they have different anxiolytic responses to morphine, which basically is just a fancy way of saying they respond to morphine differently. And we realized that humans respond to morphine in different ways. So we wanted to cover our bases and have 2 different strains of mice that respond differently. So coming back to the experimental design, like I mentioned before, the F0, the top bar, is the maternal generation. They received either saline or morphine when they were in adolescence. They lived the rest of their lives drug-free, and they were mated around postnatal day 60, which is adulthood. And the bottom line is the offspring. And the offspring were not exposed to any drugs until they were in adulthood. After a period of 5 days of being exposed to drugs, they were then immediately injected with naloxone. Naloxone is the drug name for Narcan. Most of you have probably heard about Narcan in the news. The way that Narcan works is it outcompetes opioids to sit on opioid receptors in the brain. And so once injected, it knocks all the opioids off the receptors and sends the mice into immediate withdrawal. And it happens within seconds. So mice do this really funny thing. It's called naloxone precipitated withdrawal jumping. And you can see in this video, what they do is they jump when they're when they're exposed to Narcan or naloxone. And it's a really good quantitative way of measuring their withdrawal symptoms, because we put them in these cylinders and we video them for 15 minutes and we just count how many times they jump in 15 minutes. And it's a quantitative way of showing how bad their withdrawal symptoms are. So we got from all 16 groups the data on the right-hand side of the screen, but the main takeaways from all that data is that we saw were that in general, the 129s jump less times than the C57s, which is what we expected because they're different strains. They respond differently to morphine, and it makes sense that the 129s are jumping less times than the C57s. One thing that we saw that we didn't really expect to see was that mice who were only exposed to saline during their lifetime, they never saw any morphine, they were jumping too. So that indicates that they had some sort of withdrawal symptoms. And the reason we kind of thought this was happening was because we figured that their endogenous opioid system was because they had been injected with saline, they were stressed, and so their endogenous opioid system was upregulated. And the way that Narcan or naloxone works is it knocks any opioids off the receptors, even endogenous ones, which are ones naturally produced by your body. So we figured that Narcan was just knocking off the endogenous opioid system, and they were actually seeing withdrawal effects from their own endogenous system. And we wanted to look at whether or not the C57s were jumping more times because they were actually having worse withdrawal symptoms, or if they were just jumping more times because they like to jump more. So we came up with something called the Withdrawal Score, and it's measured on a 0 to 4 scale, 0 being no withdrawal symptoms are present, 4 being all 4 are present. And the withdrawal symptoms we looked at were tail rigidity, which you can see here. It's something that mice do when they're withdrawing from morphine. You can also see some GI distress. That was another thing we looked at. We looked at something called oral stereotypy, which looks like they're chewing on the air almost. And the last thing we looked at was something called wet dog shakes. They kind of shake like a dog. And when we looked at all this data, we found that actually the 129s and the C57s had very similar withdrawal symptoms. So it wasn't that the 129s weren't withdrawing from morphine at all. They were just showing it in different ways. They weren't jumping like the C57s were. But we also saw the same thing we saw in the jumping data, that the mice who were just exposed to saline, especially the females, were also showing signs of withdrawal. So they never received any morphine during their life, and they were still showing withdrawal symptoms when given naloxone. So the reason we thought this was happening was the same as the jumping. It's because this endogenous opioid system. But the interesting thing with this data was that the C57 females who only received saline in their lifetime, but their mothers received morphine, they were showing no withdrawal symptoms. But the C57 females who only received saline in their lifetime and their mothers received saline, they were showing withdrawal symptoms. So that leads us to believe that there is some sort of transgenerational effect happening here. So the next thing we looked at was the actual opioid receptor expression in the brain. We looked at the expression in the nucleus accumbens, highlighted in purple here. And the reason we looked at it there is because the nucleus accumbens is really important in the reward pathway in the brain. The reward pathway is critical for addiction, depression, any sort of pleasure-seeking activity. So it was really relevant to what we're looking for. And the takeaways from that data were just that opioid receptor expression is higher in C57 male mice whose mothers received morphine. So if you see here, all the C57 males whose mother received morphine, regardless of whether they received morphine or saline in their lifetime, their receptor expression was higher. So C57 males, regardless of whether or not they received morphine or saline in their lifetime, but if their mothers received saline, they were just predisposed to have lower expression of the receptor in their brain. So the results of the data indicate that maternal opioid exposure prior to mating can significantly impact the health of the offspring. And these changes are likely due to those epigenetic gene by environment changes. And the implications of that, like I mentioned briefly, are that maternal opioid exposure causes changes in the reward pathway, which is super important in addiction and pleasure seeking. So it has really relevant effects to the opioid crisis, but also to its implications in this reward pathway in the brain. Thank you. And I briefly thanked Dr. Anika Tori before, but she came in, so I want to thank her again. She's amazing. She's here now, so thank you. There's a bunch of different directions this research could go. The lab I'm working in right now, they're going to expand it from just looking at the nucleus accumbens. So these mice, we took micro punches of the nucleus accumbens to look at receptor expression there, but we also took punches from the same mice in other brain regions to see if we're seeing the same effects in other brain regions. So those are the immediate first steps, and I will probably be working on that this year, so I can get back to you with the results from that. Any other questions? So because we had 16 experimental groups, we had relatively small sample sizes of 9 for each group. Any other questions?
What other assays can we do to figure out what type of effect was actually occurring, what epigenetic effect was occurring on the chemical level of the person? Would you be able to anything at that type of microscopic level?
Well, we did PCR to look for the receptor expression, and that's the best way to look for receptor expression. We could also do, if we wanted to look at the actual single cell changes, we could do single cell RNA expression assay, and from that we could see actual changes in expression, but that's very expensive.
So you tested the 129 and the 67, and I'm wondering how the differences between those two groups can be reflected, like in humans.
So we really don't know that as of right now. We know that from previous research, we know that the 129s and C57s just have different responses to morphine. And the reason we use those two is because we know that humans have different responses to morphine. So we wanted to see if this would be significant across both strains. The receptor expression that we saw was not significant across both strains. It was only shown in the C57s, but I would say we would want to repeat that with a bigger sample size to see if that is, with a bigger sample size, that result stays the same. Sure.
One more question.
Okay.
Can you potentially look at human populations to see if there's a decrease in withdrawal symptoms in particular areas of the country where you have mothers that have been exposed?
Yeah, it's something we could look at. That data is really hard to get just because anything in humans is harder to work with. And also, a lot of times that data isn't very well recorded when mothers were using opioids, when they weren't, if they were exposed in utero, if they weren't. So that data is really hard to get. It is another next step that we could try to look for in the future.